Hi all, this is Papaya with Papaya Pythons. Let's talk about the axanthic trait in ball pythons. Let's start by looking at what the word means. So xanthic is an adjective describing a yellow or yellowish color. And a is a Greek prefix meaning without. Taken together, this means without yellow. So axanthic animals have lost the ability to synthesize, transport, or properly localize yellow pigment. Today we'll focus on synthesis, but in reality, the biological pathways required to create, move, and place pigment are very complex. If you've spent any time browsing axanthic ball pythons, I'm sure you've come across the question, what line is she? There are 4 distinct lines: the VPI line, the Joliffe line, the TSK line, and the Marcus Jane line. Why do we call these distinct lines? Well, the reason is that if you take any 2 individuals and breed them together from different lines, you end up with all wild-type or normal offspring. This implies that there are at least 4 distinct ways to stop the production or proper localization of yellow pigments. Let's take a look at the molecular mechanisms behind this, but first, let's set some ground rules. Whether you're a person, or a plant, or a cute cuddly snake, most of you is created by a series of carefully controlled chemical reactions. Here's the kicker. Your DNA encodes proteins called enzymes, and these are your primary chemists. They create you. So let's say your DNA says, okay, I'm going to need 1 aluminum pot, and then it gets made. We'll call this our enzyme. So any given enzyme has a substrate it changes, or helps to change, into a product. Let's call our substrate pasta. Our enzyme's going to create a microclimate to change the pasta into something else that we want, the product, or in our case, dinner. The most important part of this is that the enzyme is unchanged by the reaction that it's facilitated. That's the definition of an enzyme. So your DNA encodes these enzymes, but it also can have changes in it, or mutations. If we change just one word, aluminum, to wax, we get a very different outcome. The same scenario leaves us with a melted pot. Just one tiny mistake or mutation can yield a completely non-functional enzyme, leaving plenty of substrate, but no product. But now let's go back to our axanthic lines. So let's say we have an enzyme pathway, and we start with one substrate. We have an enzyme that changes it into another substrate. We have another enzyme that changes it into a 3rd substrate, and another enzyme that changes it into a 4th substrate, and another enzyme that changes it into the color yellow. Now, in this pathway, having a non-functional mutation in any single gene for any single enzyme could cause an axanthic phenotype. If you knock out the first enzyme, you're going to end up with no substrate number 2, which means that even if enzyme B, C, and D are functional, they have nothing to do. They just sit around like Congress, not helping anyone. However, if you have a mutation in enzyme B, you get no substrate number 3, regardless of how many substrate number 2s are sitting around waiting. The same is true for breaking C, having nothing to give to D, and breaking D and not making yellow in that final step. Of course, it's unlikely that the yellow synthesis pathway is really that simple. Otherwise, we wouldn't see so much variation between hatchlings and adults in yellow or brown colors, or saturation. This, though, gives us a very cool example of how breeding can help us understand molecular pathways and genes. For more videos like this, subscribe and visit papayapythons.com for science you can see. Thanks all!